Move on to our next speakers. I think we're going to have a tag team of speakers here with Dr. Al George and Dr. Christopher Ahern. And so I'll introduce them both and then let them take, you can go ahead and get your slides ready if you'd like. Dr. George is a professor and chair of the Department of Pharmacology and director of the Center for Pharmacogenomics at Northwestern University School of Medicine. He also directs the NINDS-funded Channelopathy-Associated Epilepsy Research Center Without Walls, which was recently funded and is a large collaborative effort to catalog mutations and effects of those mutations in ion channels. Christopher Ahern is a professor in the Department of Molecular Physiology and Biophysics and a member of the Iowa Neuroscience Institute at University of Iowa, near and dear to my heart. I grew up near there. And he has developed a platform of gene therapy technology, engineering transfer RNAs used to correct premature termination codons. So I think we're going to hear a little from both of them in the next half hour. Thank you.
Great, thank you very much, Heather. And I want to thank the organizers, particularly Mike and Hans, for inviting me and allowing me to tag team today with my friend and colleague Chris Ahern from the University of Iowa to tell you a little bit about a conceptual approach to correcting a certain class of mutations in SYNGAP1. And these are premature stop codon mutations. And I'll give a little bit of an introduction with some background, especially for members of the audience who aren't familiar with the nuances of molecular biology. And then I'll turn it over to Chris for him to share with you his brilliant and elegant technology for rescuing stop codons, premature stop codons. And then I'll take it back for just some final words about what we're cooking up that could be applied to SYNGAP1. So let me start by just sharing with you sort of the big picture. This is what is often referred to as the central dogma of molecular biology. It's what we've come to understand is the flow of genetic information from the cell, which contains the DNA that is transcribed into RNA, and then the RNA is translated into protein. And certainly anywhere along this pathway, one can have a genetic mutation that disrupts the process and thwarts the effort to make an important biologically active protein. But today we're going to focus a lot on things that disrupt the translation step and results in incompetent proteins being made. So translation involves the reading of messenger RNA by an organelle called the ribosome. And ribosomes scan messenger RNA almost like a barcode reader would read a series of barcoded instructions. In the case of the ribosome, the barcode is actually clusters or groupings of 3 nucleotides present in the RNA sequence. And each of these 3 clusters of 3 nucleotides, if read properly in the right sequence, will code for a specific sequence of amino acids in a protein. And so the process of reading the RNA involves a specialized type of RNA called transfer RNAs that are depicted here schematically. And these are small, very small RNA molecules that are built to recognize the barcodes in messenger RNA and couple that recognition with the coupling of the molecule to an amino acid. And then the process is directed by the genetic code. Each triplet codon in the mRNA sequence, or the barcode, defines a specific amino acid. So the process of translating mRNA sequence into a protein is graphically illustrated here, where the ribosome docks to messenger RNA. It recognizes a first codon known as the start codon, which codes for methionine residues in a protein, that allows the cognate transfer RNA to dock to that position. And then the ribosome scans along and finds the next triplet spot where another transfer RNA is bound. And eventually you get a coupling of the 2 amino acids that are placed in close proximity, and the peptide synthesis begins. The process continues sequentially with as many tRNAs bound to the mRNA as possible until the ribosome reaches a signal for which there is no tRNA coupled to an amino acid, and this is called a stop codon. And so this is the natural process of generating a polypeptide. There is a class of mutations, of course, that introduce stop codons prematurely in a messenger RNA. And unlike the normal sequence of events where one reads the mRNA and generates the full-length protein, which presumably will be functional, in these disease states where there is a premature stop codon, the protein can't be made in its full length and is either made in a truncated form, which may not be biologically active, or the messenger RNA will undergo a process called nonsense mediated mRNA decay, which will disrupt it, and you get nothing. And unfortunately, in many cases, premature stop codons essentially thwart the entire translation process, and no biologically active protein is made. Now in SYNGAP1, a large fraction, maybe close to half of all the reported mutations are in fact premature stop codons. And here is simply a linear depiction of the SYNGAP protein molecule with its various domains, and shown as small colored lollipops at various locations along the protein are the various stop codon mutations that have been reported. And these stop codon mutations may involve different amino acid coding triplets in the sequence. Most commonly are glutamine to stop codon mutations and arginine to stop codon mutations. But there are representatives of very much all of the possible stop codon types. So if there was a way to repair these individual mutations, one might be able to allow the translation process to continue reading through that stop codon and not make a truncated protein, but perhaps make the full-length protein, which may hopefully be functional. Now, there's been a number of opportunities, or efforts, I should say, to develop small molecule therapeutics to try to force the ribosome to read through a stop codon. And those have worked in the cell systems, but I think they've run into trouble when they've been moved toward clinical applications. And so the read-through technology driven by small molecules has not really earned the respect of the therapeutic world. So one needs another strategy to tackle this particular type of mutation. And this is where the brilliant mind of my colleague, Chris Ahern, comes in. And so I'm going to hand the baton to Chris, and he's going to explain to you what he does after he unmutes.
Yeah, I've unmuted. Thanks, Al, for that very elegant introduction to molecular biology. And thanks as well to the organizers for letting us tag team and kind of bring to your attention this really conceptual level of a therapeutic that we're working on at this point to develop. And we're pointing it towards SYNGAP as well as a number of other proteins. So the premise for the idea that we have of using transfer RNAs, as Al discussed, involves this idea from human genetics that we now know that there are many different genes for human tRNAs, as many as 450 different tRNAs. So as Al was showing, you have a tRNA that decodes a triplet codon. There are actually many different types of tRNA that can decode those triplet codons. So for instance, with arginine, there are more than 30 different types of tRNAs, which are unique sequences, yet they all possess the commonality of decoding an arginine codon to an arginine encoding in a polypeptide. And so what we decided to do was to take advantage of that biological diversity and to construct a platform in a biological screen such that if we could audition the ability of tRNAs to tolerate mutations within this anticodon loop that we're pointing to here on the left side, if Al can run the screen. I'm not sure if he wants. Yeah, so there's an anticodon region of the transfer RNA, and this is the part that engages into the mRNA and decodes the triplet codon and then allows for the encoding of the amino acid. And so what we did is we systematically altered the anticodons of transfer RNAs such that they would no longer suppress their cognate amino acid codon, but instead suppress a triplet stop codon. And we chose the ones, the 10 families of tRNAs, which are uniquely vulnerable to conversion to stop codons. And so what we did is we changed all those systematically to stop codon suppressors, depending on what disease stop was most relevant. And then we built a screen in human embryonic kidney cells such that we had a reporter, a nanoluciferase, which is an enzyme. And that enzyme, when it's fully functional, makes a fluorescent substrate. And so what we could do is if we put a stop codon in that enzyme, we can look at the ability of transfer RNAs in human cells to rescue that stop codon by making the full-length enzyme and thus having a fluorescent output, with the idea being that it's in the right environment such that, you know, we will be able to then learn more about which tRNAs are going to be effective for this conversion. And as you may guess, many of them are actually not tolerant, but we were surprised to find that many actually are. And so we can go to the next slide. And so this is now, I know it's a data complex slide, but what you're looking at here is by color first. Each one of these individual colors are different families of transfer RNA genes. And so just for instance, in the upper left, these are glycine tRNAs. So all those bars are actually looking at the activity of those different tRNAs. And on the bottom, it's a log scale. So what you're looking at here for each one of those, in each one of these columns, there's a red dotted line, and that means there's no activity over background for those classes of transfer RNAs. But what you can see is, for instance, in the glycines, and this is a log scale, we have 3 tRNAs which successfully decode UGA stops at over 100 to 1,000-fold over background. Now, notably for SYNGAP, arginine, this family of transfer RNAs, was actually very tolerant of being converted into UGA stop codon suppressors. The ones that work versus the ones that do not work is very non-obvious. We weren't able to predict this outcome, so we really needed to do this full screen. And so when we look at this entire data set, this is now a platform of transfer RNA novel sequences. Within this data set, we have potentially a genetic repair mechanism for every known human premature termination codon within this library. And for many of these families, we have multiple different transfer RNAs that can be auditioned for their ability to rescue specific stop codons. A note on the transfer RNAs themselves, they're actually quite small. If it wasn't obvious from Al's talk, they're about 75 to 80 bases. So in terms of payload for AAV, we can fit multiple transfer RNAs within a payload. And also they don't really rely on promoters in the classical sense that we would normally be driving a gene expression. They just have a small, maybe 75 base pair leader sequence. So the whole kind of deliverable unit, as it were, is really quite compact. And so the idea of what can be delivered, we have a really good kind of buffet of genetic material that can be brought in. And what we're doing here is what we do is we would essentially convert the stop site back to its cognate amino acid and really making a wild type protein. And this will be based on the mRNA itself that's being expressed in the cell. So there's no chance for upregulation of the protein that's being expressed, which is a big thing we worry about in ion channels, which is where my main expertise is, but also that we know that we're making, at the end of the day, a wild type protein. So then the main concern that we have going forward is, what about real stops? And I'll get to that in a second. But what I'd like to just kind of show you in the meantime is, seeing is believing. And so what I'd like to share with you is just what do these transfer RNAs look like in terms of SYNGAP mutations? And so we'll go to the next slide. And so what we did is we took some of our tRNAs out of our library, and we're looking at GFP now. And so we put different stop codons into GFP, and we looked if we could rescue them. So on the left, we have an image of wild type GFP transfected into HEK cells. When you throw a stop codon in there, the cells are fine, as you can see on the left, but they have no fluorescence because GFP has been terminated. So now all we do is if we co-express one of our tRNAs from our library, in this case, we pulled out, I believe it's an arginine or is it a glutamine? So in this case, it was one of our top performing arginine tRNAs. And all we need to do is co-express this arginine tRNA, and we rescue almost near wild type levels of GFP and hex cells. And so the same can be shown. I think we have some glutamine data that we also did. Yep, and so here's some glutamine data where just all we do is we co-express. This is a different stop codon. This is the amber stop TAG or UAG in RNA language. So we can rescue this glutamine completely or really well close back to wild type levels. And so what we have here is a library or a platform to rescue. But the most important thing we need to convince ourselves about are off-target effects. Every protein, as Al showed, ends with a real stop. And we don't want to read through those real stop codons because that would cause systematic elongation of the proteome, and that would be very bad in a biological sense or therapeutic. And so what we wanted to do is we wanted to look at the read-through of real stop codons. And to do that, we turned to a method called ribosomal profiling assay. And so with ribosomal profiling, what you do is you can actually monitor where all the ribosomes, those little organelle units that Al was describing in the beginning of the talk, all those different ribosomes, they're scanning RNA throughout the entire cell, thousands of transcripts, many different ribosomes sitting on that RNA. And what you can do is if you isolate that RNA from a cell, you can then cleave away all the areas where there's no ribosomes and then sequence that little bit, that footprint where the ribosome is sitting. And then you can get an assay of where else are all these ribosomes, with the hypothesis that if we're reading through real stop codons, you'd start to see ribosomes showing up after the 3' stop. And so this is just a summary of the data from human embryonic kidney cells overexpressing a variety of different tRNA suppressors. And what you can see is this sawtooth activity is essentially the ribosome hopping down the end of these transcripts. And these are all the transcripts lined up bioinformatically from these cells. And you can see they're hopping down the last codons of their reading frames, and then they start to slow down near the stop site, and then they fall off. And in the presence of these tRNAs, you see no evidence of significant read-through of any kind at these real stop codons. And I'd love to, there's some mechanistic reason why this is taking place, we think, but I don't really have time to go into it for the purposes of brevity of this talk. We can also look at this data another way, if you go to the next slide. So this is then just, again, looking at the read-through of these 5 different high-performing tRNAs, as well as a read-through drug at a low concentration. And what we're looking at is read-through of different types of stop codons with these different types of tRNAs. And again, we really don't see much evidence statistically of read-through of real stop codons. And that really agrees with our observations in the lab, where we've never seen either if we overexpress these tRNAs in many different cell types as well, HEC cells, we're now in neurons, they're really well tolerated across the board. And so the last thing I want to be able to show you before I go back to Al is specificity of encoding, because with ion channel proteins and many genes, neuronal genes, you really need specificity. Missense mutations can be as deleterious as nonsense mutations in many instances, and that's very much true in the ion channel world as well as for SYNGAP. And in those small molecules that Al mentioned, they haven't really gained traction therapeutically. And one reason for that is they put the wrong amino acid in. So they cause this ribosomal skipping, but they introduce a missense at the stop site. And so what we wanted to do is just to take a little peek at what are these tRNAs encoding at the stop sites. And so that's what this data on the left and figure in panel A is. This is mass spec data. This is basically, we can purify a protein, a test protein in this case, out of a HEC cell that's been rescued. And we can ask whether or not, in one case, we put a glycine in, that's the middle panel on the left, or a tryptophan, and the masses work out to be identical to what you would expect for correct encoding. This is now an area that we're going into, but the fidelity of the tRNA, the ability to suppress the right amino acid and put the right amino acid back where it's supposed to be, is intact. So that's a very important aspect, but that's something that will need to be tested on a case-by-case basis as we go look at different genes. But it looks like the fidelity is there. We're not hitting real stops, and we're also getting the right amino acid in. If we look at panel B here, what we've been able to do is just look at, are we hitting the right stop codon, right? So we put different stop codons in this luciferase gene, and when we recode this glycine tRNA to suppress UGAs, it suppresses UGAs. And then in panel C, when we compare this to some of these read-through drugs, even if you can guess that they're working maybe a little bit, so we have gentamicin there, we have a little bit of rescue, G418 is an experimental compound, but then even compared to these tRNAs, it's not even close. So the tRNAs do a much better job of suppressing, they put the right amino acid in. And so as in terms of like a biological vector, we think they have a lot of promise for stop codons, because as Al was mentioning, there's a lot of different, positions architecturally within SYNGAP. They're all different. They have all different contexts. They're all different amino acids. And we don't necessarily need to completely understand the complex molecular genetics and physiology of SYNGAP to know that they all share a common mechanistic basis for the dysfunction. And we see this as a potential way to at least start rescuing some of these stop codons. And so with that, I'll go back to Al.
Thank you, Chris. So I think just in the last couple of minutes, I'll just share with you kind of the vision that we have going forward to apply this technology to SYNGAP1, the premature stop codon. So again, I showed this before. This is simply a linear map of the protein showing its various domains, decorated by the location of various stop codons. The two that we think would be most valuable to target first would be the arginine to stop and the glutamine to stop, given that they represent more than 65% of all known SYNGAP1 mutation, premature mutation types in the gene. So we envision producing 8 different SYNGAP mutations, 4 arginine and 4 glutamine to stop codons, express them in hex cells, and then use the tRNA technology to see if we can't rescue them. And the readouts that we've envisioned will be simply, can we restore the synthesis of full-length SYNGAP protein in the cells, using immunoblots to assess that. And if that's possible, maybe we can also demonstrate using an enzymatic activity assay, the restoration of RAS-GAP enzymatic activity. And then as Chris highlighted, it's important to demonstrate the fidelity of the repair, that the stop codon has been rescued with the correct amino acid. And for that, proteomic analysis can be done. And then lastly, to make sure that the native stop codon isn't read through, this ribosome footprinting strategy is what we have in mind. These are in vitro experiments in heterologous cells. The natural next step would be to use also heterologous in vitro experiments, but to use neurons. And so to derive human neurons, as Dr. Kova had explained, one can use iPS cell technology, which we do routinely in my lab to generate both cortical excitatory and cortical inhibitory neurons. And so we've imagined generating these neurons from patient-derived iPS cells that have premature stop codon mutations in SYNGAP1. And I'm grateful to some members of the field who've actually offered to share with us some of the existing cell lines that would be valuable in this regard. Chris's lab engineers adeno-associated virus with multiplexed recoded tRNAs that can be engineered to go into neurons. And then again, the neurons could be interrogated with immunoblot strategies to see if full-length SYNGAP protein's been restored, maybe functionally, and also importantly, to look at neuron morphology and culture with and without the rescue tRNA. Then last but not least, of course, if the in vitro experiments were successful, one would want to go in vivo. And so for this, we would have to engineer a mouse with the corresponding premature stop codon mutations. None of these are in existence at the moment. And then if that was available, we'd certainly want to try to do in vivo delivery using AAV viral technology to deliver the tRNAs to the mouse brain, and again, assess restoration of SYNGAP active protein and maybe enzymatic activity if possible. Ultimately, the goal long-term is perhaps to try to demonstrate behavioral rescue in mutant mice. And so that's a long-term goal, which I think we're not going to be there anytime soon. But with that, I just want to thank people in my lab that are queued up and already actively working on this technology applied to ion channels in collaboration with Dr. Ahern, and to acknowledge Dr. Ahern's team and also some collaborators that we've engaged with recently to try to get some of this work funded. So with that, I again thank the organizers and thank you for your attention.
Great, thanks to both of you. I think that's actually really exciting. We have a thank you, not a question, in the Q&A at the moment. Hans had a question in the chat, actually. It said, would it be useful, a useful study to determine which premature truncation codons would be targeted for NMD and which would avoid NMD? Or do we have data that tells us something about that?
Yeah, so I think, there's sort of a general dogma that NMD is engaged for premature stop codons throughout most of the coding region of most genes, except when you get into the last exon and you're very close to the tail end of the protein coding region. And so that's the general dogma. Certainly one would want more specific, gene-specific information before tackling this project, and I think that would be something that would be worth doing. Right now, I don't have any of that information at my disposal, but it's a good question.
Yeah, but and also just to add to what Al was saying, and also for to address the question, is it's our working hypothesis at the moment that the activity of these engineered transfer RNAs would actually antagonize nonsense-mediated decay, because we think that they're going to interfere with the regulation and the clientele of NMD proteins that already recognize those stops. And so by having this tRNA present, we're hoping that actually will increase the amount of mRNA that's present as well. But again, that's something that we don't have access to any of that information right now. And so for us, the SYNGAP is really a great test protein to start to look at that. Because as Al said, there's a dogma out there, but I think we're in a position to start to really test that. Yeah.
Yep. Okay, two quick questions before we move on. Thanks for the, let's see. Clever and intriguing strategy. I'm hoping you can talk a bit more about why there isn't more read-through of real stops. Isn't there concern that read-through of even a small subset of targets could have important biological consequences?
Yeah, sure. So read-through happens actually normally. The body is equipped to deal with read-through that happens at a few percent all the time under normal situations. So there's a couple different reasons why read-through doesn't happen. That's because the premature termination codon is in a really different environment in terms of the nucleotides that surround it compared to the evolutionary footprint of a real stop codon, which itself has a clientele of proteins, these stop codon binding proteins, EF1 and EF1, the release factors that bind to real stop codons and derail ribosomal translation. Premature termination codons don't have that, and so we think that they're much more vulnerable to tRNA suppression. Real stops have a whole protection mechanism built in that keeps read-through from happening. But even though read-through happens at a small level, most cells are equipped to deal with that small amount of stress. So we're hoping that we can hit the sweet spot between those two mechanisms, and currently it seems like we're able to hit that window. Again, everything is a therapeutic window. So, that will come down to testing this platform that we have. We have multiple different tRNAs for any family. So we can then sample if there are ones that do tend to read through at real stops more than others. We haven't seen that. But if we do, we have other ones we can fall back on. So we're looking forward to getting into that work. Absolutely.
Great. And then last question from Janie. Do you predict that the PTVs will have the same phenotypes in mouse? It's surprising there aren't these patient type models yet. And then she notes that her son has a single nucleotide deletion resulting in a stop without a frame shift. And is it possible to engineer a compensatory frame shift into the tRNA?
Yes, I can tackle that. I mean, certainly it is, it's not that surprising that there's not a premature stop codon mouse model at this point. I think to model haploinsufficiency of SYNGAP1, it was logical to do just a global knockout of the gene, and that I think has been done. So I think that's not that surprising. The other part of the question is about the frame shift, and unfortunately, frame shift mutations disrupt the ribosome's ability to capture the right barcodes because the reading frame has been distorted. This technology that we talked about today would not correct that. And I don't believe that it would be valuable to correct a stop codon that's in the wrong reading frame, because you might read through and make a protein sequence that's not natural. So this technology would only work for in-frame premature stop codon mutations.
Yeah. Great. Thank you both. I think that is a really intriguing study and a hopeful step forward for SYNGAP1 potential therapy. Yeah.